Good afternoon to everybody, and welcome to the TMS seminary. Today the seminary is about chemometrics, a multifaceted discipline, and the speaker of today is Alessandra Biancolillo. She obtained her PhD degree in spectroscopy and chemometrics by the University of Copenhagen. She has been a postdoc researcher at the University of Rome La Sapienza, at the ERSTEA Institute in Montpellier, and currently she is a researcher at the University of l'Aquila. Her research field is chemometrics. She has worked both on method development and on the application of chemometrics in various fields. So if you are ready, Alessandra, you can start and enjoy the seminary.
Thank you, Sarah, for the introduction. So today's seminar is on chemometrics, which is a branch of analytical chemistry finalized to data analysis. This is an overview of the seminar. At the beginning, I will introduce you chemometrics. Then I will give you a brief description of one of the most popular chemometric method, principal component analysis. And then we will see two examples where we will apply chemometric method onto real dataset. In one case, we will apply principal component analysis on an archaeological dataset, and in the second example, we will apply classification method on a clinical dataset. At the end of the presentation, I will give you some general conclusion on the application of chemometrics in the various fields. So I know that all of you belong to different fields of science, but despite this, I think that when we all are planning our research study, we all go through the same steps. So at the beginning, we have to investigate the state of the art. So we have to investigate the background of our research topic. We will go into the literature to see what has been done, what were the conclusions drawn by the different scientists, and we will focus on what has not been done yet, obviously. So after the investigation of the state of the art, we will need to plan our experiment. In chemistry, usually we do it by design of the experiment, but I know that this can be applied also in other fields like biology or physics. Once we have planned our experiment, we will have to carry out our experiment. So we will do it in our lab, maybe we will use some instrument, or maybe we can do it by our computer, just making some theoretical calculation. Regardless the way we carry out our experiment, at the end, we will always have to analyze the outcome of our experiment. So at this point, chemometrics jumps in. Chemometrics has been developed in order to analyze chemical data, but despite the way it was conceived, actually it is used to analyze the data obtained by any kind of experiment. So we can use it even in different fields than chemistry. So if we search for chemometrics in Google, we will see that it is often associated to the words that you will see here. So maybe you haven't heard about chemometrics before, but I'm pretty sure you've heard about some of these words like multivariate statistics, pattern recognition, machine learning, metabolomics, and so on. So chemometrics, as I said, is a branch of the analytical chemistry. It's a relatively new discipline. This guy here, Svante Vold, is considered his father, and he defined chemometrics as the art of extracting chemically relevant information from data produced in chemical experiments. So chemometrics is used to analyze data obtained by experiment, and its method, so chemometric method, lay on other disciplines, so they lay on statistics, mathematics, and informatics. If we search for chemometrics in Scopus, we will see that half of the publications are related to chemistry, but at the same time, we will see that there are publications related to other fields, like biochemistry, agricultural science, physics, pharmacology, environmental science, and so on. This is related to the fact that we can find in the literature a lot of application of chemometrics, which is used to solve problems in different fields. But if you look at this graph, we will see that at the same time, a lot of publications are related to other science like computer science and mathematics. This is because despite all the application we can find, there is still a lot of effort put in developing novel chemometric method, and this is related to the fact this discipline is relatively new. In fact, chemometrics born in the 70s, and if you, like you can see here, where I have reported the publication per year from the 70s to nowadays, we see that chemometrics has attained attention, in particular in the last 30 or 40 years. This is mainly related to two principal reasons. The first one is that nowadays it's easier to have a personal computer. Almost anyone has a laptop, and moreover, these computers are more powerful today. So the calculation that we do today, and they take maybe some minutes, would have taken hours in the 80s. Another reason why chemometrics has been attentioned in the last years is because also new instrumentation that we have in our lab are more advanced. So instrumentation are able to take into account different sources of variability at the same time, and they produce bigger and more complex data. So we really need multivariate statistics to analyze this kind of outcome, because otherwise we would lose some information, and this means that somehow we are vanifying all the effort we put in the lab. So, but what really chemometrics can do for us? Let's imagine we have a set of samples, we analyze it, we will do it in the lab, we will take some measure, I don't know. Then, by means of chemometrics, we can take three different paths. The first one is the one leading to exploratory analysis. So, we apply exploratory analysis when we want to investigate our data without any a priori knowledge. This means that in this way we can see whether our data has special trends or suspicious trends, if samples have the tendency of grouping, or if there are some outliers. We can see all these things by exploratory analysis. There are different methods for exploratory analysis. One is principal component analysis, and this is the method that we'll see soon. After this, a second path we can take by chemometrics is the one leading to regression. I'm pretty sure you have heard about regression before. There are different chemometric methods for regression. The most famous, I would say, the most commonly used is partial least square, PLS. This method are used when, for example, we want to quantify compounds in our sample. Chemometric regression methods are particularly useful when we want to quantify analytes in presence of interference, or when we are doing it in complex matrices. Eventually, the last step we can take by chemometrics is the one leading to classification. When we classify samples, what we want to do is to predict their class membership. This means that we want to divide our samples according to specific characteristics on the basis of their characteristics. This probably sounds a bit obscure, but we will see it better later in the last application I will show you. So now it's time to see exploratory analysis. We will see a bit of theory behind it. Exploratory analysis is a step that we will probably always take when we applied chemometric analysis. So even if our final goal is to solve a regression problem, before applying a regression model, we will probably use an exploratory analysis method in order to see whether there is something strange in our data. So if we want to investigate the behavior of our data. Before talking about the theory behind principal component analysis, we need to understand how we go from our lab to our computer. So let's consider the case where we have collected some signals on our samples. For instance, here I have reported some near-infrared spectra. So we export our signal from the software of the instrument to the computer. And then in the computer, we will have to create a data matrix. Here I call the data matrix X. This matrix will have as many rows as the number of samples that we have investigated, and as many columns as the number of the variables that we have collected. In this case, we collected spectral variables, but this is just an example. Then, if we want to apply principal component analysis, what we will do is a bilinear decomposition of the data matrix X. So X is decomposed into a principal component matrix, T, and a loading matrix, P, plus E, which is the residual matrix, where is all the information which has not been modeled. So from this, we realize that principal component analysis is nothing than an orthogonal projection of X onto the space spanned by the loadings. If we look at the equation on this slide, we see that principal components are linear combinations of the original variables. So we pass by having original variables to having latent variables, which are called principal component. The first principal component is the linear combination with the maximum variance. The other principal component that will be extracted will be calculated with the same reasoning, but under the constraint that they have to be orthogonal with respect to the previously extracted components. Nevertheless, I don't want you really to focus on the mathematics behind the extraction of the principal component. What I really want you to understand is that when we are extracting principal components, we are somehow compressing the information present in the X data matrix into a reduced set of variables. So we will pass by having the X data matrix, which has dimensions n times I, to having a matrix, the principal component matrix T, of dimensions n times V, where V is way smaller than I. So if we come back to the example of the spectral data, for instance, collecting near-infrared spectra, we collect approximately 3,000 original variables. Then, if we apply principal component on this data matrix, we will extract a smaller number of principal components. There are some methods in order to optimize the number of the principal component that have to be extracted. I'm not going to talk about this today. But regardless the way we do it, we will have a number of principal components. So this means that we pass by having 3,000 variables to having 6-7 variables. The fact that we are working with a reduced set of variables where the information is compressed allows us having some graphical tools. One of the most useful of these tools is coarse plot, and is a plot like the one shown here. So we can realize a score plot projecting our samples onto the space spanned by the first principal component. This means that in this way we can have plots, 2- or 3-dimensional scatter plots, where to investigate our sample. In this plot, we will observe the behavior of our sample. We can see if, like here, we can recognize some grouping tendency. We can see if there are outliers or if something in general is strange in our data. Now, probably these things will become more easy looking at the practical application of principal component. So in this case, we use principal component analysis to investigate some archaeological samples. This study came out because some archaeologists came to us bringing some samples. As you see, they had unearthed some human bones from two different necropolises. The necropolis were one in Italy and one in Sudan. So these bones belong to people belonging to different cultures in different ages. We had different bones. We had ribs and vertebrae. What the archaeologists asked us was whether it was possible to develop a method which would help them understanding the age of these bones, because sometimes they don't have a clue about it. So what we did at the beginning was to use a non-destructive method to analyze these findings. So we used near-infrared spectroscopy equipped with an integrating sphere, and we collected NIR spectra on this. And then we destroy a very small part of the samples in order to carry out thermogravimetric analysis. After we collected our data, we create a principal component analysis model on this data. So this is the scores plot that we obtain. I don't know if you can see some trends, some groups in this plot. We actually did. So we recognize one group in the top left part of this plot, one group in the top right part of this plot, and one group in the center. So then we came back to the archaeologists, we showed them this plot. Obviously, we gave them the labels of these samples, and they provided us the information about the age, because they are actually able to understand the age of some findings, looking at the tones or looking at the cracks on the bones. The problem is when they are not sure about their age. So once we included the information they provided us into the plot, we realized that we were actually able to see differences among the samples according to their age. So this is very important, because this plot that you see here means that it is possible to use this approach to discriminate the age of the samples. This means that if the archaeologists now unearth a new sample and they have no clue about the age of this finding, we can analyze this sample with the same analytical techniques we have used, so NIR and thermogravimetry. Then we can apply this principal component model, and we will see where the new sample fall in this plot. So for instance, if the new sample will fall here in the top right of this plot, we will say, okay, probably these samples belongs to the 3000 BC. So we will be able to give a tentative age of this sample. In the case we want to be sure about the origin, about the age of these samples, what we will have to do is to apply a classification method. This requires that, in that case, we have some other samples whose age is quite sure. So in this case, using principal component analysis, which is an explorative method and not a classification method, we can just give a tentative idea of what is the age of the sample. So now we can discuss the second example, where we will see a bit better how classification can help us. So, in order to give a general definition of classification, we say that we are classifying when we divide samples according to characteristics on the basis of the, we classify samples according to categories on the basis of the characteristics samples present. This is probably not very clear, so I have shown here an example just to let you understand what classification is. So imagine you have a bag of apples, you have red and green apple, and you want to use a classification method in order to divide these samples according to their color. So we will take this apple, we will analyze them, we will apply a classification method, and the method will tell us, okay, this apple, this sample is red, so it belongs to class red. On the other end, on a green apple, the method will say, okay, this apple is green, so it will belong to class green. I know that as long as we can discriminate the green and red color, we don't really need a classification method for doing it. That's obvious. This is just a trivial example in order to let you understanding what classification is and what a classification method can do. But now let's see a second example. Let's consider the case we have a bag of yellow apples and we want to know which apple has been harvested in Italy and which apple has been harvested in England, for instance. So in this case, the origin of these apples that look all more or less the same is not something that we can predict at first sight. So we cannot look at an apple and say, okay, this is an Italian apple, because the origin affects chemical characteristics of the apple which are not visible. So in this case, if we want to trace the origin of this apple, we will need classification methods. We will actually need a set of apples harvested in Italy, surely harvested in Italy, a set of apples surely harvested in England, and then we will apply our classification method on new samples. So once we have trained our method, our model, we will be able to make predictions on new samples. Classification is commonly used in different fields. For instance, it's used in industry for process monitoring. It's used to realize if we have samples in lots which are strange. Then it's used in quality control, as in the example that I just mentioned. It's used for traceability. This is very important in food analysis, in particular when we have high added value products like PDO food, for instance. Classification is also used for detecting frauds like adulteration and counterfeits. This is once again useful in food analysis, but is also used in pharmacology, because unfortunately there are still on the market a lot of counterfeit drugs. So these are usually detected by classification method. And eventually, classification is also used in case control studies to solve problems related to clinical trials, as in the one that I'm going to show you. So in this second study, we have investigated a physical medical condition called physical frailty and sarcopenia. This is a geriatric condition which can affect people older than 70 years. This person observes a muscular mass loss and they have problem in working without devices. So when a person goes to the doctor complaining about these symptoms, what the doctors do is usually ask this person to do some physical test. So the person will be asked to make the walking test. So the person is asked to walk for 500 meters in less than 15 minutes without any device. Then the second test the person will do is the sitting-standing test. In this case, the person is asked to sit and stand rapidly in a short period of time. And eventually, the strength of this person will be measured by specific devices. At the end of all these physical tests, the doctor will calculate a short physical performance battery. This is an index, is a value, and according to this value, the doctor will say this person is sarcopenic or this person is not sarcopenic. So the doctor complains about the fact this index is actually not very accurate. So that's why they came to us asking for help, because they do not have any clinical test to predict this medical condition. So they came asking us whether it's possible to define new biomarkers for this medical condition. So we took part at this study. We had 200 participants. Obviously, all of them were geriatric patients. They took the physical test. The doctor calculated the shared physical performance battery on all these persons, and according to these values, they divided the participants into cases, so sarcopenic persons, and non-sarcopenic persons, controls. So we had 100 of sarcopenic participants and 100 of non-sarcopenic participants. Blood samples were took on all the participants. In particular, we investigated in the serum cytokines, inflammatory markers, amino acids, and a set of further proteins. So eventually we had 4 different blocks of data for a total of 74 variables. So at this point we decided that a suitable classification method for the analysis of this dataset was a method called sequential and orthogonalized covariant selection discriminant analysis. This method has a bit long name, so we will call it SEO-COPS-LDA, as it would be long to explain you the algorithm of this method, so I'm not going to doing it, also because I don't think it's really in the scope of this seminar. What I really want you to understand here is that this method is a classification method, so it will tell us which participant is sarcopenic and which one is not. Obviously, it will do it on the basis of the blood samples taken on the participants. And at the same time, together with the classification, the model will tell us which are the most relevant variables for the solution of this classification problem. So this means that the model will tell us which analytes are characteristic of each category. Category case and category controls. So once again, this means that the model will tell us which analyte in the serum are characteristic of the sarcopenic condition. So this will be the novel biomarkers we can use. So as I said, we collected data, we applied the SEO-COPS-LDA, and then we classified participants in cases and controls. As I said, at the same time, the model selected for us 7 variables, 2 cytokines, 3 inflammatory markers, 1 amino acid, and 1 protein. So for what concerns the classification prediction, we had a good agreement with the classification made on the basis of the short physical performance battery index. So the agreement was of the 80%, which is definitely good. And then we came back to the doctor showing them which variables were selected by the model. So they were quite happy with this selection, because in their opinion it makes sense that this variable are related to sarcopenic condition. We selected 2 inflammatory cytokines, which are generally lower in concentration in the serum of the sarcopenic patients, and this is related to the fact they have problem in the regeneration of muscles. Then we selected 3 markers of inflammation, asparagine, aspartic acid, and citrulline, which are related to the synthesis of glutamine. These analytes are generally higher in concentration in the serum of the cases, and these indicate a perturbation in the metabolism of glutamine. Eventually, we selected other 2 analytes, which were lower in concentration in the serum of the sarcopenic patient, and these are related to oxidative and metabolic stress. So, this study is still going on. It's a European project, and actually they are using these biomarkers to predict the sarcopenic condition, and we are quite happy with it because they are having very good and promising results. So now I will draw some general conclusion on chemometrics and on its application. So what would you like to remember from this seminar is that you have to take in your mind that data are always more complex than you expect. Sometimes we are looking for something in data, and for this reason we are focused on something and we don't see what else is in our data. So that's why I think it's important to use data analysis method, because otherwise we may lose some information, and this means that somehow we are vanifying all the effort we put in carrying out our experiment. So what I always suggest to students and other researchers is to apply chemometrics, study chemometrics or ask for help, but apply it, because secondary characteristics could be less feasible, but this does not mean they are less relevant. So at the end of our, we need to put a bit more of effort, but we have to perform data analysis, because we will always discover something more, and in this way, this means that we will always gain something. So it will require a bit more of effort, but it will pay back. So at this point, I would like to thank you for your attention. I hope you found this seminar interesting, and well, thank you.
Thank you, Alessandra, for your nice presentation. Very interesting and clear. And now it's the time for questions. I can read for you the questions in the chat. There is one. Is the PCA only a graphical method? Can we associate the graphical output to a number that describes its significance or the strength of the associations, like a p-value?
Okay, well, PCA is not only a graphical method, in the sense that I show you just a graphical tool that you can use for investigating your data, but there are several others. For instance, it's also very important to investigate the loading matrix, because the loadings will maintain the chemical meaning of your experiment. So for instance, you can have an idea of which variables are contributing the most to the model, so which one are the most relevant. You don't have a p-value. If you want, probably this question was put before I talk about classification, probably if you want to have an idea of the significance of what you are looking into, you will better use a classification method. So that will tell you, for instance, if you are classifying, what's the accuracy of your classification, so that will give you a number.
Okay, thank you. And another question, could this type of analysis used in drug discovery, in particular during the first screening of a chemical library?
Okay, sorry. Well, it depends on your aim. So it depends on it depends on what you want to see. So, for instance, the first screening of a chemical library, I don't know, if you want to see, for instance, if some of the drugs you are investigating have similarities or dissimilarities with some others, you could use exploratory analysis. yes. So in that case, you can do it. You can use it on data found in the literature, for sure.
Okay. And is it possible to use this approach also as predictive tools, for example, to foresee a result of an experiment changing a variable such as temperature, concentration, solvent, and so on?
Yes, definitely. It's a very useful application. This is very important in order to see what's the effect when you change something during your experiment. Yes, for sure.
Okay. Are there more questions? Don't be shy to ask. Just a curiosity. What is the chemometric error rate? What could be due to any errors in the classification?
The error rate depends. Every time we create a classification model, for instance, we will have an error rate, and that depends a lot on different parameters. So it will depend on our dataset, obviously, because as I said, we need to train our model. So this means that we need some samples whose class belonging is sure. So the first problems could be that we are using samples that are not appropriate to train our sample. Another problem is when the class we are trying to model has a too high variance. So when inside the class the samples are too different one from each other. So the highest part of the error rates we obtain creating classification method is generally related to the nature of the data. Then obviously we will have errors related to the errors linked to the experiment and so on. Moreover, sometimes the error could be related to the choice of the classification method, because we have different, and sometimes in some situation one is more appropriate than another. So maybe we took the wrong method and we can have some artifacts. So we have problem then in making predictions. All of these methods require the optimization of mathematical parameters. So also in that case, we can make something that is wrong, and so the final error will increase. There are several reasons why the error could be high, but in general, it's possible to work on it and to obtain very good error rates.
Okay. So more curiosities? If not, I have two curiosities too. One is, if you have any kind of a data set and you don't know the connections, for example, you can always try to apply this method to search unknown connections or grouping or some features in common, or not?
Yes, you can do it. If you use principal component analysis, you don't need any a priori information. So you can apply it, and then you can investigate scores plot or other kind of plots that will tell you whether you have connection between samples or experiment, or even if you have connection in the variables, you can investigate different aspects, but you don't need a prior information.
Okay, thank you. And another curiosity is about the hardware. You talked about the necessity of a computer. Mm-hmm. Is necessary a particular hardware to obtain consistent and solid results, or not?
No. In general, I put in the slide. I didn't talk about it. In general, when we make chemometric calculation, we usually use some software like R or MATLAB. But often in the software of the instrument, there are some chemometric methods that are implemented. Like, for instance, PCA is often implemented in different software. I think also in Excel. I'm not very used to Excel, but I think Excel does it. So a normal laptop, if you don't have to run a very complex calculation, you can use a normal computer, no problem.
Okay, thank you. We have another question. So, if I understand correctly, the signatures of markers, biomarkers you generate are meant to be used in diagnostics, but also as a method to discover variables that are relevant to the condition you are studying. Is it possible to use a non-homogeneous dataset in the latter examples to find out connection between gains, grip, strength, blood glucose, BMI, just random examples?
Yes, it is. In that case, actually, the method that I mentioned, SEO-Copsel DA, is one method. It belongs to a family of methods that is called multi-block method, and they have been realized in particular to combine block of data that are of different nature. So it doesn't matter if you have BMI or gene information, because, for instance, you can categorize your gene information in a categorical way, like in a binary way, for instance, and then you can have another block where you have continuous variables, and it doesn't matter. The model will be able to base its classification on these data blocks. So yes, it's possible.
Okay, thank you. I don't know if there are more questions. If not, I will send you in the chat a form. Okay, I'm sending you. And you should fill this form. There are 3 questions. In order to improve the quality and the understanding of this seminary, of this TMS seminaries, and in order to improve the comprehension, the liking, the interest. So just 3 questions. You are invited to fill the form. So if there aren't any curiosities, I can another time thanks to Alessandra for the nice presentation and for the availability. And I can introduce you the seminar of next Thursday. The title is Something That Everybody Should Know About Optical Communications by Cristian Antonelli from the Department of Physical and Chemical Science of this university. So that's all, I think. Thank you for the participation, and see you next Tuesday.